So our idea here is, how do we make a trustworthy, reliable, efficient lab in the middle of nowhere? And so we've been doing this in the Amazon River. I joined a project where there's the Los Amigos Conservation Concession that's on the Madre de Dios River. And this is in the southeastern corner of Peru. And so this conservation concession has been there for, I think, 20 plus years. And it's bordered by the Madre de Dios, which is heavily mined, and the Los Amigos River, which actually has a ranger station at the base to help protect it from mining. And then the biological station itself is pretty self-sufficient. You know, there's dormitories, there's a commodore, and you get fed, and there's a kitchen, and then laboratory space for various projects. And you can kind of rent it as a researcher, or you can go as a tourist and do a lot of birding and that sort of thing. And this beautiful network of trails that branch off of it. So there has been some of a lab there, but with this new funding from the Moore Grant and this in situ laboratory initiative, what we're doing is really building up a molecular lab. So we have the genomic side of things. We have 2 opentrons, we have a MinION, we have PCR. So in theory, you know, these samples can go straight from the field where we're doing the marker capture programs, collecting hair, feathers, tissues, whatever, and instantly get extracted, amplified, tested, and sequenced right there. And that's just absolutely fascinating from a biodiversity standpoint. You know, we'll get to the point where we can do eDNA, we can swab a paw print and figure out who's walking by where.
What's eDNA?
Environmental DNA.
Thank you.
So just the trace amounts of DNA in the soil that you would assume, like little pieces of skin from the paw left behind, or like a fecal sample or something like that. So we're building up this biodiversity side, who's out there, who's related to who, what's going on. Right. And then we're building up this pathogen ecology side. So what pathogens are in their blood, viral screens, bacterial screens, ectoparasite screens, that sort of thing. And some of that is initially going to be very targeted. And then hopefully we're going to branch into families of viruses and that sort of thing. If there's a hit, you know, we can narrow it down. Yeah. And then we have the part that I'm like, wait, this project's cool and all, but like, are there any other concerns? If you have an animal and it has this lesion and you find this virus, can you really make that connection that this virus is causing that lesion? Or do we need to think about anything else going on? And it turns out, like I said, massive gold mining, which produces lots of mercury. That's part of the process that they use to pull out the gold, because mercury and gold bind each other. They can get these glumps and they burn off the mercury. So it goes into the air and goes everywhere. Not great. And it's obviously a really big accumulator. And mercury exposure has been studied a lot, but it's one of those things where like, well, what happens in tropical systems? What happens in amphibians? What happens in snakes? What does that mean? You know, like when you start to ask those questions, you realize there's a lack of data there. So what we're building is this ability to test mercury at the site alongside all these other pathogen type screenings and biodiversity screens, and take this data set, which is going to be collected yearly across biota, bats, birds, non-human primates, tamarins specifically, small to medium-sized animals, so everything from like opossums and squirrels to anteaters and sloths and things like that, and monitor over time to see if things that are happening in our world through climate change, through changes from deforestation and mining, like do those things have an impact on these animals? And there's many layers here. There's an animal movement ecology layer that's being built into there. So it's going to be this fascinating long-term cross biota study that will hopefully start to give like a lot of evidence and oomph to like, we need to be doing something about this. It's not just we tagged 6 collars or 6 vulture collars and we saw that all 6 went in different ways. Like, what do we do with that? Right. I don't know.